Hi class, how are you? So this is actually my second attempt at today's lecture because I forgot to turn the microphone on. And I hope the second run through here is going to actually be as good as the first, because that was actually one of my better things. It's funny how when you don't have the microphone on. But today we're going to cover mitochondrial import and energetics. So we'll basically look at a lot of sort of the mitochondria physiology and specifically protein import into the mitochondria in the first part, and then in the second part we're going to look at energetics. So just some background on the mitochondria. And cells need energy to survive and grow and function and divide and just do their normal day-to-day activity. And the largest source of energy in a cell is actually ATP. And we've seen lots of different enzymes that use ATP for energy. And the vast majority of the ATP is actually made in the mitochondria. There is some that's made in the cytosol, but it's a small amount relative to what's made in the mitochondria. Mitochondria are actually kind of cool because they have different compartments, and they have two lipid bilayers. And one of the neat things about that is that it creates a unique problem for sorting, because certain things have to go to different compartments in the mitochondria. But what's really nice about the two lipid bilayers is that it's actually really ideally suited for energy conversion. So the proton pump driving ATP synthesis, so in oxidative phosphorylation. And we'll cover that in the second part of the class, or the second lecture today. And energy conversion in plants actually occurs in chloroplasts, and there's a lot of similarities. I'm covering the chloroplast today just because if there's anyone interested in it, I personally like it a lot, photosynthesis and stuff, so I'm going to cover it. And also there's a lot of similarities with how things are transported and done in the mitochondria, so it just is a reinforcing thing. I won't test you on anything that with plants, but it's a good thing to know. Maybe some of you are thinking of going to graduate school and doing something in the plant biology field, so it's useful to know. Okay, so we've already covered a lot of this. We've seen this schematic before, and we looked at all the proteins that are sort of translated in the cytosol, and then they get transported to various organelles, the endoplasmic reticulum, and then we looked at transport to the Golgi and the later secretory pathway. We also saw how proteins that are translated in the cytosol are imported into the nucleus and the peroxisomes. And today we're going to look at how proteins that were sort of synthesized in the cytosol get to the mitochondria and to the plastids in plants, and chloroplast in particular. So here's just sort of the structure of the mitochondria. And you can see that there's technically four compartments here, right? There's an outer membrane, and then there's an inner membrane, and then an inner membrane space, and then the matrix. And so a protein that's synthesized in the cytosol will need to actually be transported to the right location in the mitochondria. So that represents kind of a challenge, and it's a little bit more complicated than what we saw for translocation into the ER. But it's really sort of just a fascinating area. And if we look at chloroplast, it's somewhat similar. There's an outer membrane, an inner membrane space, but then there's also the stroma, and then you have the thylakoid membrane, and then the thylakoid space. So depending on the plant protein and where it needs to go in the chloroplast, there's also different mechanisms for sorting. But the sorting is somewhat similar between the two. So here's a grade EM of a mitochondria. We actually saw this on the first day of class. You can see here the matrix, and then you can see the inner mitochondrial membrane and the inner membrane space that's between them. You can sort of see it right in here, and then the outer membrane. And so these different compartments actually represent sort of import challenges, and we'll look at how different proteins get transported to different regions in the proteins that are involved. Here's just another EM of a mitochondria, and if you sort of do sort of a 3D modeling of it, this is what the three-dimensional structure is. So you can see the inner mitochondrial membrane actually sort of weaves back and forth, and this creates a lot of surface area for oxidative phosphorylation to occur. Now, as with transport into the endoplasmic reticulum, transport into the mitochondria requires a mitochondrial import sequence or a signal sequence for a mitochondrial protein. Here's just an example of one, and that's going to basically form an alpha helix, and then that is bound by a receptor that brings it into the mitochondria. And so these proteins get fully synthesized on the ribosome, and then they have this mitochondrial import sequence, and then that's a sort of a signal to bring it to the mitochondria and translocate it into the mitochondria. And then it gets, depending on the sequences that are in the signal sequence, it will get shuttled to the right compartment in the mitochondria. And here's the proteins that basically do it, somewhat similar to what happens in the endoplasmic reticulum in translocation, but also significantly more complicated. But it's a little easy to remember if you sort of realize that the names have great acronyms. Okay, so there's the TOM complex, which is transport outer membrane. So it's just an acronym, TOM, transport outer membrane. Okay, and that has the receptor that's going to basically recognize the mitochondrial import sequence, and then there's a translocation channel. Okay, there's also the SAM complex over here, which is sorting and assembly machinery, SAM. Then on the inner membrane, you actually have the TIM complex for translocase inner membrane. There's the TIM23 and the TIM22 complex, and then there's also the OXA complex. Okay, so, and this OXA complex, as you'll see in a second, is involved in genes that are basically mitochondrial encoded genes. The proteins that they make get inserted into the inner mitochondrial membrane through the OXA complex. But one of the things, for the vast majority of proteins that go into the mitochondria, the TIM and the TOM complex sort of function together, as we'll see in a second here. Now, one of the tools that they've used to sort of study mitochondrial import is this really sort of great translocation assay. And so you can in vitro transcribe and translate a mitochondrial protein and then mix it with purified mitochondria. And if you sort of mix that on ice, what ends up happening is the in vitro transcribed and translated protein will sort of get partially inserted, and the signal sequence, the mitochondrial import sequence, will get cleaved off. And then if you take this that's at sort of 4 degrees Celsius and you treat it with protease, the vast majority of the protein will get cleaved up. However, if you take this where it's sort of stalled at 4 degrees, partially imported, and you just sort of incubate your purified mitochondria at 25 degrees, so room temperature or so, it'll import the protein, and then it becomes protease resistant. If you add detergents and proteases, that'll actually solubilize the membrane, and then the protein actually gets cleaved up. So using this type of assay in combination with mutant mitochondria and the TIM and the TOM complex, they've really been able to sort of work out exactly the mechanisms of import and export. Okay, and they can do that with energy chelators and things like that. You can also fractionate the mitochondria into its different compartments, so you can basically take your purified mitochondria and basically put it in sort of a low osmolarity thing, and that will rupture the outer membrane, but the inner membrane sort of stays intact. Then you can sort of centrifuge it out and separate the outer membrane and inner membrane proteins, and then you can take your sort of inner membrane and then once again do solubilize it and separate things out, the outer membrane, the matrix, and the inner membrane. Then you can see what proteins are where And this type of assay was also used to sort of figure out where different proteins go in the mitochondria when they're looking at translocation, and also how the transport's affected if you were to do this sort of assay here with some of the various mutants that they had. Right, and transport into the mitochondria is somewhat essential. Certain organisms can get away without having a mitochondria, certain eukaryotic organisms, but in general, they use this, and you can get temperature-sensitive mutants in the TIM and the TOM complex. So, okay, so based on all of that and the sort of experiments where they have a in vitro transcribed and translated mitochondrial protein with a signal sequence, and they can add it in. It actually binds to a receptor on the TOM complex, and then it gets inserted through the TOM and also sort of co-translationally inserted through the TIM, not co-translationally, but basically translocated through TOM and TIM sort of simultaneously. TIM and TOM can work together, and then it gets translocated into the matrix, and the signal sequence is cleaved, and the protein becomes folded. Okay, so TIM and TOM typically work together, but they can also function independently in this process. The energetics of import. So getting back to this too, one of the things that definitely understand how this whole process works. Here, I want you guys to sort of understand it, but there's more energetics to it. So normally, once a protein is being sort of translated in the cytosol, if it's a mitochondrial protein, it's bound by cytosolic HSP70 proteins. These are chaperones. They're ATPases. And they'll help guide the protein to the receptor on the TOM complex. Okay, and then using the sort of the ATP hydrolysis, it'll start to sort of feed it in, and then once it's in, you have a membrane potential on the in the inner mitochondrial membrane, and this is partially due to the electron transport chain, and then that membrane potential will actually insert it into the TIM23 complex. Okay, and meanwhile you still have the cytosolic HSP70 proteins sort of threading the protein through the TOM complex. Okay, and then once it's sort of in the TIM complex, it will, the signal sequence will be cleaved, and then the mitochondrial HSP70 proteins, also ATPases, will sort of grab hold of it and sort of ratchet it in, not too dissimilar from what we saw with BIP in the endoplasmic reticulum, as well as in bacterial transport when we're looking at bacterial translocation, and the SEC-YEG complex. And SEC-A was the ATPase in that instance that threaded it through. So very sort of similar mechanisms. You have a cytosolic HSP utilizing ATP to drive the protein through the outer membrane, and then on the inner membrane in the matrix you have a mitochondrial HSP70 that's utilizing ATP to pull it in. Now, different proteins are brought to different parts of the mitochondria. So let's say you have a nuclear encoded mitochondrial outer membrane protein. Well, that'll get recognized. It'll be brought in through the TOM complex, and then there's chaperones in the inner membrane space, and they can then pass it off to the SAM complex, and it'll become fully folded protein in the outer membrane. And mitochondrial porins sort of follow this pathway to the outer membrane. You can also get proteins to the inner membrane space in the inner mitochondrial membrane via a couple different mechanisms. So you'll once again go through the TOM and the TIM23 complex, and it'll have a couple of signal sequences. The first one that guides it in will be cleaved, and then the second one is a signal sequence, sort of a stop transfer, that tells it's an inner mitochondrial membrane protein, and then the translocation will just stop and it gets released. You can also have inner membrane protein brought completely in through the TIM and the TOM complex, and then the signal sequence is cleaved, and then there's a second signal sequence that actually sends it to the OXA complex and inserts it into the inner mitochondrial membrane. Likewise, if it's a mitochondrial protein, and we'll look at some of the mitochondrial genes in a second that go this route, but you'll have mitochondrial protein synthesis, and then that will have the signal sequence, and the OXA complex actually will insert it into the inner mitochondrial membrane. You can also have proteins in the inner membrane space where you can actually get it cleaved off, and then it's just an inner membrane space protein. Or you can have mechanisms where something goes through TOM, it's bound by chaperones, and then passed to the TIM22 complex using the membrane potential, and then it becomes an intermembrane protein. So like I said, it's a bit complicated on how it does it. You don't necessarily have to memorize every single step here. Definitely know this process, but here just sort of be cognizant that depending on where the protein is coming from, different signal sequences will guide it. And that's sort of the requirement of all of the different translocation machinery that are on the mitochondrial outer membrane, as well as on the mitochondrial inner membrane, right? So in the inner membrane, there's TIM23, TIM22, and the OXA complex, okay? So transport into the chloroplast is really similar. You have a chloroplast signal sequence, and then it'll sometimes, if it's headed to the thylakoid space, it'll have a second signal sequence. Goes through translocators, sort of similar to TIM and TOM, and then when it reaches the stroma, the first signal sequence will get cleaved, and then you have the exposed thylakoid signal sequence, and then it'll get imported into the thylakoid space or into the thylakoid membrane, depending on the signals that are there. And it goes into the thylakoid space using the thylakoid transport machinery, and there's a number of different ones. There's ones that uses ATP and the electrochemical gradient. This is very similar to the SEC pathway, which we covered in bacteria. If you guys go back to when we covered translocation into the endoplasmic reticulum and SEC61 complex and stuff like that, there was the SEC-YEG. It's very similar here. There's an SRP-like pathway where you have a signal recognition particle protein, very much like SRP, and that gets inserted. And then there's a TAT pathway, which we haven't covered TAT, but it's also common in bacteria. And then there's a spontaneous one where you supposedly get spontaneous insertion. However, there's probably a translocation machinery here that they just haven't figured out yet. Because as we know, proteins don't necessarily just like to spontaneously translocate into the membrane. Okay, mitochondria are also very sort of plastic, right? They can change their shapes. They're very dynamic. They can actually fuse. They can fissure off and just change shapes. And here's just a little schematic to try to show you how dynamic mitochondria are, right? It's not just this one static thing. And as we've sort of seen over the course of the term, that, just in general, cells are very dynamic. And so here's just a good example of how you can go from a sort of mitochondria. It can fissure off into small pieces, and then they can actually fuse back together again. And if you look at this mitochondria right here, sort of in a time lapse, you can start to see that it changes in certain areas, fissure off. I think if we follow this, it sort of fissures. There you can see the fissure. Now it's getting bigger. And then you can also see other things that sort of fissure off or fuse together. So if you're sort of looking here, you can see that this all of a sudden will fuse right there, making this connection. And there it is again. And then it'll eventually break apart again. So the point is, you can look at this all day if you guys want, but and find things where it's coming together and fusing. But the big take-home message is it can fissure into small pieces and it can fuse, right? And if part of the mitochondria gets damaged, it can undergo autophagy and get recycled and then remade. And so they can sort of grow in size and be made larger. Here's just another schematic of the fission. And you can see it sort of breaking apart, and then if you go back in the other direction, it fuses. And here's the mitochondrial DNA that's in here. We'll cover that in just a second. Here's an EM of mitochondrial fission. Looks to me like it could almost be fusion too. It's really hard to say just by looking at a static image, but you get the idea how it's sort of coming apart or coming together right here. And somewhat similar to almost cell division looking. So one of the things with mitochondria is they tend to co-localize with microtubules. That gives them a nice little attachment area. And so here's just staining for mitochondria here in this cell. And then if you stain the microtubules here, you can see that basically the mitochondria are co-localized to the microtubules. And that's pretty easy to sort of fathom if you guys consider that, all the microtubule motors, the kinesins, they're going to be using a lot of ATP and, other things with the microtubules, the cytoplasmic dynein and whatnot. So that's where a lot of the energy sources is. And it gives them a good place to anchor into the cell too. To help keep them localized. So muscle cells actually have a ton of mitochondria, right? Because you have all of the myosin 2 that's going to need ATP to actually move on the actin in the sarcomere, and so excuse me, I got a little tickle in my throat. Sorry guys, got a little tickle in my throat here. I just ate a peanut granola bar before I started lecture, so sorry. But here you can see the mitochondria. They're actually in and around the sarcomere. This is just a sort of schematic of cardiac muscle. And you can think of all the myosin 2 that needs ATP. So you're going to want to have a large supply of ATP, especially in a muscle like the heart, right? Because it's just beating all day. You also can find a lot of mitochondria associated with the sperm tail, right? The sperm needs a lot of energy to move, and so all of the mitochondria is sort of wrapping the flagella there. There's also DNA in the mitochondria, so here's just another image of mitochondria, and sort of spotted in sort of yellow here is mitochondrial DNA. And so ultimately, proteins come from 2 locations into the mitochondria. You can have genomic encoded mitochondrial proteins, and then the mRNA is transcribed, and then it gets out to the cytosol where it's translated. Then it becomes a precursor protein with the mitochondrial import sequence, and then it's imported via the mechanism we just saw. But you can also have proteins that are encoded by the mitochondria in the mitochondrial DNA, and that makes RNA, and then the proteins are synthesized actually in the mitochondria itself, and then it gets transported to the correct location. And we saw how that happened with the OXA complex, right? And a lot of the proteins involved in electron transport in the electron transport chain are actually encoded by the mitochondrial DNA. Not all, but some. Okay. Here's just a schematic of the relative sizes. Here's the human relative to some other organisms, and you can see the human is actually really small. And if you look at, and we covered this before, this is the evolution of the mitochondria. Here you have your sort of early anaerobic prokaryotic cell. It's got the DNA that gets packaged into a nucleus, and then aerobic prokaryote basically gets engulfed, and then there's this great symbiotic relationship and they start to share things where you have the mitochondria and the nucleus. So here's just some of the genes and how they overlap depending on the organism. The humans shown here in sort of pink, and then you have Schizosaccharomyces here. So there's a couple common genes that are in basically all mitochondria. Cytochrome oxidase is one, so COX-3. Here's just the human mitochondria. So you have, of course, the 16S and the 12S ribosomal RNAs, right? They're going to make the mitochondrial ribosomes. And then there's cytochrome B, which is part of the cytochrome BC1 complex. You have the NAD hydrogenase subunits. There's some over here, a couple over here. Subunits of the ATP synthase, and then cytochrome oxidase subunits, right? That's the COX-1 and COX-3 that we saw over here, the cytochrome oxidase. So that's it for part 1. I will start part 2 in a second. And sorry about my little coughing fit there. I'm going to go fill up my water before I start part 2. Okay? I'll see you in a bit.